Good day, this is Professor Will, CBMD PhD. And today's Wednesday, January 19, 2022. It is 2:55 and 38 seconds, a.m. Eastern Standard Time. Today's research, antibiotic resistance. So let's hop to it here. Halting antibiotic resistance is a little less futile. Date January 18, 2022. Source, Rice University. Bioscientists developed a microfluidic platform for high-throughput studies of how bacteria evolve antibiotic resistance. The idea of using microfluidics to encapsulate cells isn't new. There are plenty of lab-on-a-chip devices already. What the researchers done is apply it to biomarker discovery for antibiotic resistance. The established technique for studying resistance involves feeding antibiotics and gradually increasing amounts to vials of bacteria in a solution. That reveals how some microbes evolve resistance, but favors mutants that grow fastest and makes it difficult to control other factors, like population size, number of generations, and the space they occupy, all of which can influence the evolutionary process. Putting cells into microdroplets overcomes those limitations by allowing researchers to fine-tune their environments, allowing them to study new evolutionary trajectories. The droplets are a controlled environment where the cells grow in their own ecosystems. That is similar to what happens when a wound gets established in your body, where there are a few bacteria instead of the millions in the test tube. Researchers say these microfluidic droplets are a better application of the early stages of an infection. 1 millimeter of bacteria doxycycline emulsion can contain about 2.6 million droplets, each of which can be thought of as a tiny bioreactor. It only takes about a teaspoon of the solution to quickly produce literally about 1 billion microdroplets, whereas before we might have used liters. And this allows researchers to do densely replicated studies that just aren't possible when you are limited to test tubes. Researchers control the size of the droplets through microchannels etched on the silicone wafers through photolithography. The etched wafers were then used as masters to mold the polymer devices. For their proof-of-concept experiments, they exposed single-cell Escherichia coli bacteria to varying amounts of deoxycycline to analyze the conditions under which bacteria evolve resistance to the common antibiotic. And each experimental run contained one configuration of droplet size, bacteria count, and antibiotic concentration. And after allowing the cells to reproduce, their genomes were analyzed. The droplets are fairly sturdy because of the surfactants researchers use and maintain their coherency for over 24 hours. Researchers can then extract and isolate the DNA and send it out for whole genome sequencing. The researchers noted their platform turned up not only the kind of evolution seen in traditional studies, but also previously unknown pathways to resistance that involve large-scale alterations to cells' genome structures. And with their initial experiments done, the lab is now testing a range of microbes to see how they evolve resistance. Researchers are expanding the zoo to include other more difficult pathogens, including a strain of E. coli they know is quite dangerous in hospitals. The researchers say the platform may also be employed to quickly and accurately analyze biomarkers and screen the drug candidates, the topic of ongoing studies in the laboratory. Good day, this is Professor Will, CBMD PhD, on microbiology, bioinformatics, and pathophysiology. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.